Hi everyone and welcome back to my channel. In today's video, we'll be discussing the key technique in the plant molecular biology or the plant biotechnology, that is freeze-thaw method for Agrobacterium transformation. As we know, Agrobacterium is a gram-negative soil-borne pathogenic bacteria that has unique ability to transfer its part of the DNA, that is called tDNA, into the plant cells. The Agrobacterium has a natural ability. So we have hijacked this natural ability of the Agrobacterium to transfer our desired genes into the plant cells, and that's why we are getting the transgenic plants with having more disease resistance capacity, as well as having more crop productivity. So now I have cloned my gene of interest into the plasmid vector. It is a binary vector, especially it's a binary vector. And now I wanted to transform this plasmid vector into the Agrobacterium cells. Now this Agrobacterium will infect the plant, and whatever the tDNA region is there in my plasmid, that will be transferred into the plant cell. So this is the first step for going into the plant transformation, that I have to transform my constructs, that is the plasmid vectors in which I have cloned my gene of interest, into the Agrobacterium. And now after that, the Agrobacterium will infect the plants and then transfer the tDNA region of that vector. So today's video will be discussing, or I will show you how to carry out the freeze-thaw method for Agrobacterium transformation. So I have competent cells already prepared, and I'm just thawing it on the ice for 2 to 5 minutes. Now label the competent cell vials in order to identify in which cell we have transformed what. After labeling the cells inside the laminar airflow, I am adding the plasmid DNA. Here I am adding 1 to 2 microgram of plasmid into the competent cells. Make sure you are adding inside the cells and not on the walls of the tube. And after adding the plasmid, you just gently flick the tubes and keep it in ice for at least 5 to 10 minutes. After incubation on ice for 5 minutes, we need to freeze the cells in the liquid nitrogen for 5 minutes. Make sure you are wearing all the protective gears. Freezing disrupts the bacterial cell membrane and creates transient pores. After freezing the cells in the liquid nitrogen, immediately we have to take out the cells, and we have to incubate in the water bath for 5 minutes at 37 degrees Celsius. So the transient pores which are created on the bacterial cell membrane are now able to take up DNA after thawing, and this is the rationale behind the freeze and thaw method. After incubating at 37 degrees Celsius, we have to transfer the vials on the ice for at least 2 to 5 minutes, and after that we will add LB broth into it. Now from past 10 to 15 minutes, bacteria is undergoing lot of stress. Especially we have freeze the cells that disrupts the membrane. Again we have thawed cells at 37 degrees Celsius for 5 minutes, making uptake of the DNA. Now to give a bacteria a chance or to recover from this kind of stress, we need to add LB broth, and we have to keep it for at least 2 to 3 hours at 28 degrees Celsius in case of Agrobacterium tumescens or rhizogenes, uh, to allow them to recover from the bacterial stress, as well as to express their antibiotic genes. You don't need to change the tip if you are not touching it. If you touch the vials of the end off, then you have to change the tip. After adding LB broth, we need to keep it at 28 degrees Celsius for 2 to 3 hours in order to recover the bacterial cell membrane from stress, as well as to express their antibiotic genes. Now after 2-3 hours we will take out the vials. Now we have to plate down the cells at 4000 rpm for 5 minutes. Now, after plating down the cells, we need to discard the upper supernatant, which is approximately 700, because I have added 800 microliters of the LB broth. Now we will remove the 700 microliters of the Luria Bartani supernatant, and we will keep the 100 microliter of LB, and we will resuspend the cells in that 100 microliter of LB broth, and then we will plate it on this the appropriate antibiotic. So this is how looks after resuspending the supernatant with cells. Now in this case my plasmid has kanamycin resistance, and the bacteria has the chloramphenicol resistance, so that's why I am plating this culture on the chloramphenicol and kanamycin plate. Spread with a sterile spreader in the laminar airflow to avoid any kind of contamination. And after spreading the culture, then make sure you are sealing the plates with a paraffin or phytara, and keep it again at 28 degrees Celsius for 2 days. And after 2 days you observe the colonies on the plate, and those colonies you can further screen by the colony piece. Thank you for watching this video. If you have any query, please comment down below. I will try to solve it. Please do like, share and subscribe my channel.